Hello, my name is Trina, and today I have come up with video on life cycle of Taenia solium. And what happens is male and female germ cell will fertilize to form zygote, and around this zygote, vital area will secrete yolk cells, and these yolk cell will impart yellowish color to around this zygote. And what happened later on is that this outer yolk cell will form chorionic membrane, and after forming chorionic membrane around this zygote, this is going to be called capsule. And all the yolk nuclei will transfer into upper part of this capsule, and this part is going to be called syncytial yolk mass. Syncytial is the word that we use to any cell which is having multinucleated cell or multinucleated epithelium, where the nuclei are not compartmentalized. Till here we have our early development stage. Early development stage completes here, and after that what happens is this capsule is going to get into the uterus. And I have told you about the Malick's gland that are present around the oviduct, which will secrete some lubricants for the to make the passage of this capsule into the uterus feasible. So initially your uterus is like this, a slender structure, which is a slender straight structure, and this can't occupy more capsule. So in the gravid proglotter, this uterus is going to get branched to increase the surface area and to increase the storage capacity of this capsule inside it. And now in the uterus, the further development takes place. Cleavage occurs, and cleavage is holoblastic and unequal. What is holoblastic? Holoblastic means the whole zygote is going to get cleaved into two parts, and unequal means two unequal cells are going to get generated, and those two unequal cells are, smaller one is embryonic cell and the larger one is megamer, and syncytial yolk mass as it is. Now what happens is this embryonic cell, this will divide into two types of cell: smaller micromer and larger mesomer. And this megamer are going to show mitosis and will divide into a lot of megamers, and this megamers later on form an outer embryonic membrane, and this micromer, they are later on going to make form the morula, and this mesomer, they are going to form the embryophore. At this stage, the syncytial yolk mass, this is actually nutritive in function, and they will transfer they will actually provide nutrition to these megamers. Now what happens is, as I've already told you, that micromer, they have formed morula here, and we have our mesomer have formed our embryophore here, and megamer, they have formed outer embryonic membrane, and this outer embryonic membrane is nutritive in function and will provide nourishment to the whole embryo, and later on this disappears. And now what happens is that this morula is going to get transform into hexacanth. Some cells of this morula will show will start forming some hooks-like structure. Those are 3 pairs of hooks-like structure, and this stage of morula is going to be called hexacanth. And now after the formation of this hexacanth, the outer cell of this morula will secrete 2 layers, which is going to be called oncospheral membrane, and the embryophore will divide into 2 membranes, 2 membrane embryophore, and also there will be present this eggshell will also be secreted. And now this stage is called where you have a hexacanth plus 2 oncospheral membrane plus 2 embryophore plus eggshell. This stage is going to be called oncophore larva, and this oncophore larva is infective in secondary host. Secondary host in this case is pig. What happens is, inside human, the gravid proglotter of Taenia solium is going to show apolysis, and it will detach from from that tail, and will come out with the feces, and the pig will ingest those feces, take up those feces, feed on those feces, and will get infected. Inside pig, this oncophoral larva, the oncophoral membrane will dissolve. Also, the eggshell will dissolve with the help of enzyme, that is pepsin, and hexacanth will come out. Here we have hexacanth, 3 pair of hooks, as you can see. This hexacanth, with the help of its hooks, will penetrate the gut, and then it will enter into the blood circulation of pig, first into the blood vessel or lymph vessel, and then into the blood capillaries. Through the blood capillaries, it will settle down into this striated muscle, where it will absorb water from the outside and will form a bladder wall and will start invagination like this. And this stage is going to be called bladder worm or cysticerous larva. When this cysticerous larva will be in the stage of forming proscolex. Proscolex means when this invagination occurs, the larva forms scolex in the inverted position, inverted direction. And this stage is infectious in human being, which is our primary host. Actually, this is wrong. Here we have primary host. Primary hosts are the host where actually your pathogen or any kind your pathogen will show sexual development, will undergo sexual development, or the fertilization, where fertilization takes place. Now this is proscolex stage, and this is infective in humans. And when human ingest when human take up those pork which are not properly cooked, those are called measly pork, and they have this cysticerous larva present in them. And when they take up this measly pork, the cysticerous larva become active in the gut of humans. And in the gut, when they become active, they show evagination, and this inverted proscolex will come to the outer surface and will form scolex, and then neck, and then strobilize it is, and this will form a proper taenia worm, and which will be like this. This is your taenia worm with the help of okay, so here we have scolex part. Scolex is made up of rostellum. Around the rostellum we have hooks. They are generally 28 in number, and here in this around this head part we have suckers. These suckers help the taenia worm to attach to our intestinal wall. And here we have neck and the long strobila part, and each segment is going to be called proglotid, and this one is gravid proglotid. It will show apolysis and will come out with feces. This initial proglotid will show cross fertilization with gravid proglotid. So this taenia will form in humans, and it will cause 2 types of diseases in human. So first one is teniasis. Teniasis is not that deadly, and patient will show some symptom like acute weakness or anemia due to the indigestion or eosinophilia. Okay, and the very deadly disease that it can also causes cysticercosis. What happens in case of cysticercosis is that the oncophore larva, despite actually entering into the secondary host, that is pig, it will enter into the human host. When human actually take up food, generally when they work in field and doesn't wash their hand prior to eating food, there is every possibility that they will ingest this oncophore larva in the human, and this oncophore larva will take this human as intermediary host. The later on development of the larva occurs in human, and it can invade the eye muscle and cause blindness, or it can invade the brain and cause brain necrosis, which eventually will cause epilepsy, paralysis, or can cause death. So this one stage is deadly. We are having some anti-helminth drugs as well, which are camoquine, carbon tetrachloride, antifen, and dichlorofen. Usually these drugs are unable to detach this collox from the infected organs, so surgery is required. And prophylaxis are some preventive measures you can take is proper cooking of the pork before taking it, and second is personal hygiene. That's it. Thank you.